Hello everyone, welcome back to another class on regeneration biology. So in today's class, we will learn about what are the common cellular events during regeneration, embryonic development and cancer. I'm sure all of you would have heard about what are um, the events that occur during cancer, or what are the events that occur during regeneration, what are the events that occur during embryonic development. We have seen in multiple occasions in the same course itself, and we haven't covered embryonic development or cancer. But be assured that during regeneration, quite a lot of pathways that are turned on during embryonic development or turned on during cancerous condition is reactivated. So we will try to understand what are the pathways that are we can think of that has got in common. So one of the transcription factor which is induced, which is playing pivotal role during cancer development and regeneration, is OCT4. That is one of the Yamanaka factors which is induced during development very early, and it's also hijacked by cancer, regenerating tissues, etc. So importance of OCT4 in various pathways of embryonic development, regeneration and cancer is being worked out in hundreds of papers. One interesting thing about OCT4 is OCT4's concentration. What is the level of OCT4 protein present in a tissue can decide what is the role it is being doing. It is just like, if I want to give an analogy, if your wallet is strong or thick wallet, you have lots of money, that will decide where are you going to have your dinner. If your wallet has only 100 rupee, you will try to have some snacks on the roadside. But if your wallet has got 10,000 rupees, you will not have just a snack. You will go to a good restaurant and have a nice meal. Same logic applies to a lot of transcription factors. OCT4 is one of them. So in simplistic sense, OCT4 level, when it builds up to a certain level, it is having one effect, and it goes further, it is going to have the opposite effect. So I will not discuss too much, except I will touch up some how it is contributing to the regeneration, cancer and developmental biology. OCT4 is one of the pluripotency inducing factors. What does it mean? It can convert a fibroblast into stem cell. That is what it means. Somatic cells can be reprogrammed to a pluripotent state by ectopic expression of transcription factors such as OCT4, SOX2, KLF and MYC. They are, in short form, they are called Yamanaka factors, and the scientist who got Nobel Prize for his discovery. So research studies have implicated the importance of OCT4 in human zygotic genome activation. What is zygotic genome activation? During development, an egg and a sperm fertilize and you end up getting a zygote. So until few hours of development, first division, second division, third division, fourth division, all this show is done by maternal deposit. That means the RNA and protein that is deposited in the oocyte is the one which is contributing to the development initial stages until around morula stage. In the case of zebrafish, roughly around 3 hours of development. It is not going to activate a zygotic specific gene expression. Whatever present until then in the oocyte is the one which is running the show. That is the development. So this zygotic genome activation is very important because the sperm comes with a completely imprinted, completely silenced gene. So that has to be reactivated. Otherwise, paternal genes will not be. For example, I will tell you, human IGF and IGF receptor is a classic example. If maternal copy of IGF is present, a normal copy, that is not going to express in the zygote. It is going to be imprinted, it is silenced. The same way, IGF receptor is paternal copy is not going to get expressed. So no matter whether you have IGF and IGF receptor from both the parents, only the IGF from the father will be turned on and the IGF receptor from the mother will be turned on. The other will be, the opposite will be silenced. Although the father had both the copies functional, mother had both the copies functional, and they contribute also both the genes they will contribute, but the zygote it will specify. So when you say zygote genome activation, it also means the silenced one should be turned on and turned on one should be turned off also. Okay. So the research studies have implicated the importance of OCT4 in human zygotic genome activation. Epigenetic modulation of OCT4 gene and post-translational modifications of OCT4 protein activity are seen in the context of different types of cancers as well. So what cancers are doing? They are trying to recapitulate the embryonic property or the zygotic genome activation property of OCT4 is being hijacked by the cancerous tissues so that it can turn off any genes at will which will favor the cancerous cell. So OCT4 induction is essential during zebrafish retina regeneration as well. If you knock it down, there is no regeneration. There is a research publication from our own group. It's been available for those who are interested to read. So common events during development, organ regeneration and cancer. Let us look further. Throughout embryogenesis, cells bud off from developing tissues and move to shape and pattern the complex architecture of prospective organs. During embryonic development, there is too much of movement of the cells, which is essential because an organism is being formed from the zygote. So if the cells are not moving, there is no different germ layers like ectoderm, endoderm, mesoderm, etc. will not be formed properly unless the cells are moving. So a similar process occurs in adult life during wound healing and tissue repair when lingering cells migrate into injury sites to recreate the pre-existing structures. During regeneration also, it is very much similar to that of development where the cells have to move. So in the case of injury, it has to move into the spot where there is an injury so that it can colonize and restore the normalcy. So acquiring cell motility is necessary but not sufficient. Motility is needed but not sufficient for this event. It's like you got admission in a good college, you have to go to that college, but that doesn't mean that you will get the degree. You need to move, cell has to move and then behave in a certain way. Credit goes to its gene expression events in that cell after reaching that particular spot. So cells that detach from their neighbors must elude a process called anoikis, which is a form of apoptotic cell death that occurs when cells lose their adhesion to the extracellular matrix. Whenever a cell that is moved from its comfort zone, say a given tissue had 100 cells, so one cell decided to move as a part of being proliferating or a part of migrating, etc. But the moment it loses its contact, normally every cell will have around 6 to 7 contact points, of which 2 or 3 points are lost itself is good enough. That cell is now vulnerable for apoptosis, and that process is called anoikis, but it has to resist. If it's just like, you are not ready to move away from your home and you need always support from the parents, then you move out, you will perish. But you have to, during that process, you should develop. Although I am dependent on parents as of now, but I should be self-reliant. I should be standing on my own foot. Then only I can run the show. Cell also have to do this. It has to resist this matrix detachment related anoikis, which is a form of apoptotic cell death. Moreover, the migratory cells undergo extensive mitotic divisions to produce founder populations which settle in a newly forming organs during development. So these migrating cells during development also occupying in a particular area. Say like your liver is formed in a particular place, your lung is formed in a particular place, your kidneys are formed in a particular place, your heart is formed in a particular place. So this all happens because of the migrating cells, and then they differentiate not randomly according to the demand of that morphogenetic gradient what is present in that location. Same logic applies when you are talking about cancer metastasis, or same logic applies when you're talking about a wound healing. So during development, an organ is being formed. During regeneration, a new tissue is being formed in that lost area, and they must colonize. After moving, the newly formed cells colonize worn tissues during the repair. So the typical phases of embryogenesis and organ regeneration strongly resemble the pathological processes of Tumor invasiveness. Tumor invasiveness, you may have heard about it. Like many times, if anyone in your non-circle gets some cancer, doctors will say, oh, there is a secondary tumor or there is a metastasis. This word they use it. This is because you have a tumor cell in a particular location, but it goes to secondary in a different tissue. That time the cancer becomes complex, and that is called a tumor invasiveness, or we call it as an invasive tumor or invasive cancer. Similarly to cells at the wound edge. So the tumor invasion happens, the cells in the wound edge. Think about a lake. You want to close that lake. How will you close the lake? You will start putting sand or a mud in the surrounding periphery, and you keep walking into the middle of the lake. That's how. You don't randomly keep throwing mud or sand into the middle of the lake. Usually you don't do it. Same way, the periphery of the wound site, the cells has to migrate and cell has to proliferate, and they are walking into the center of the wound. This is what usually happens. And into the wounded cells at the tumor's leading front disrupt intercellular contracts and infiltrate the adjacent surroundings, where they resist anoikis, that is a form of apoptosis, and grow before lodging in the blood vessels for systemic dissemination. So when a tumor is growing, and normally it happens when doctors take biopsy also, that is also brings in an orifice for these tumor cells to come into the bloodstream, and once they get into the bloodstream, they are now looking for a new colony to establish. Often the cancer cells resurrect the latent schemes of cellular reorganization that usually are confined to embryonic development. Resurrect means they reinvent, rediscover that properties that embryonic cell had, because embryonic cell is very sturdy, have lots of good gene expression that facilitate proliferation and migration and differentiation. This is what the cancer cells also make use of it, and the damaged adult organs and leverage them to become competent for metastasization. That is, the act of metastasis is called metastasization. So these activities, that is basically the motility, survival means resisting this apoptosis, and proliferation that occur during developing and also during injury response can be either regeneration or it can be wound healing, doesn't matter. And neoplastic tissues. Neoplastic tissue is the migratory cancer cells. They are called neoplastic tissues, embody a biological program defined as invasive growth. So that means invasive growth is there during development, invasive growth is there during regeneration and wound healing, and invasive growth is there during neoplasia or the cancer metastasis also. So this property is must. This is triggered by extracellular stimuli that regulate the activity of several transcription factors that in turn modulate the expression of many other proteins ranging from cytoskeletal and cell-cell junctional components to cell cycle regulators and anti-apoptotic effectors. That means the cell should have the ability to proliferate, and the cell should have the ability to migrate, and the cell should also have the ability not to undergo apoptosis. Then it is a fit show or a fit cell. One major environmental inducer of invasive growth is hepatocyte growth factor, HGF. We have seen this during liver regeneration, and also known as a scatter factor. The ligand for the MET, that is a tyrosine kinase receptor. MET stands for mesenchymal to epithelial transition. So MET tyrosine kinase receptor, and it's also known as HGF receptor. So HGF can bind onto MET, which is a receptor tyrosine kinase, which is it can do a kinase activity also, but it can bind to the ligand HGF also. MET function is required for various morphogenetic events in both embryonic and adult life, and it drives malignant progression of several types of tumor. It is a necessary stuff, but during cancer it goes haywire. For this purpose, MET propagates an intricate system of signaling cascades that result in the comprehensive rewiring of gene expression, because the MET, when it binds to the HGF, lots of gene expression events, changes happen inside the cell. HGF is secreted as a single chain inert precursor and is converted into two-chain functional heterodimer by extracellular proteases. Proteases are induced due to inflammation or injury, etc., and they contribute in the proper maturation of HGF. Pro-inflammatory cytokines such as interleukin 1, interleukin 6, tumor necrosis factor alpha, also known as TNF alpha, and transforming growth factor beta, TGF beta, induce transcriptional upregulation of both HGF. HGF is produced because of this inflammation in fibroblast and resident macrophages. Macrophages are the phagocytic cells, they are in the immune system, and also fibroblasts are the glue cells which is present pretty much everywhere. And MET, that will not only produce this HGF, but it's also is produced MET in epithelial cells, so that the ligand is there, receptor is there. Now they have to act and the gene expression events start. Following HGF binding, the kinase activity of MET is switched on by receptor dimerization and transphosphorylation of two catalytic tyrosine residues, that is tyrosine 1234 and tyrosine 1235 within the kinase activation loop. That is the first event that occurs. The subsequent step is the phosphorylation of two additional docking tyrosinase in the carboxy terminal tail, that is tyrosine 1349 and 1356. So we have got a total 4 tyrosine in picture. When phosphorylated, these tyrosine act as a degenerate motif for the recruitment of many signal relay molecule. That means they can act as a mediator for passing on the signal, somewhat like a intermediate, somewhat as a something like a facilitator. MET is also substrate for several protein tyrosine phosphatase known as PTPs, protein tyrosine phosphatase, including the receptor PTP and density enhanced phosphatase 1, that is also known as DEP1, and it has another name, that is PTPRJ, because in different articles when you read you will also see this name, and also a leukocyte common antigen related. This is another protein which is contributed by these signaling event. HGF to MET action triggers these sets of events, so that now the cell turns on a bunch of genes. So now let us see what is the signal cooperation between MET pathway components. So as you can see here, this is the plasma membrane where the receptors are located, and HGF ligand is seen here and the MET receptor is seen here. And this will trigger a bunch of internal action or internal signaling events. As you can see here, names of these proteins are not necessary for you to remember or understand, but this picture shows how complex these signaling events are that is occurring. So this is inside the cell and this is outside the cell. Once the ligand bind onto HGF, bind onto the receptor, you can see lots of related pathway. In one scenario, you are seeing this alpha beta of this alpha 6 beta 4 integrin comes into picture, and they are coming together because of this interaction. In another scenario, same HGF binding to the MET, these proteins are not there, but you got a different set of proteins which is part of CD44V6 protein that came into picture. In another scenario, what happens? You also have this MET bound by another protein called SEMA, SEMA, and then the class B plexin comes into dimerization. So you have different types of scenario that is occurring with the same, it's all with the same kind of ligand receptor. It's almost like a Swiss army knife, and based on which tissue you are talking about, this can have a differential behavior. This is what you should keep in your mind. Not that it's just like if you say you are in India, you will read a Hindi newspaper, or you are in US, you may not get a Hindi newspaper, rather you will get a newspaper which is made of English language. Or if you are in France, you may find a newspaper which is made French. So what is the difference? Everywhere you are reading newspaper, provided you know the language, but you cannot expect this particular thing be there in this place. Same logic you should have based on which tissue. But the event is newspaper being delivered at your residence. Same way, HGF and binding onto the MET. What action will happen depends on which tissue you are talking about or which scenario you are talking about. So one good signaling event, that is good signaling, what I mean is well-studied signaling event, is Hedgehog signaling, which is important during prostate development, regeneration and cancer. So you may have seen one of the very deadly cancer is prostate cancer, which affects lots of males, especially in their post-middle age time. So let us try to understand what is the contribution of Hedgehog signaling. Hedgehog signaling in the developing prostate involves. So we are trying to understand during development what the signaling will do, during maintenance, homeostasis, what the signaling will do, during cancer, what the signaling will do. Are they doing the same through all these three phases? Hedgehog signaling in the developing prostate involves both autocrine and paracrine signaling. Autocrine means cell is releasing hedgehog signaling and acting on that cell itself. That means you are cooking food for yourself. Paracrine signaling means like your mother is cooking food. She don't make just food for you. Mother makes food for herself and also to the family members. This is paracrine signaling. And both hedgehog can do. And remember, hedgehog is a molecule that has got a migratory ability up to 3 millimeter. One of the longest distance it can pass. Because of its credit goes to its lipid moiety added onto it, etc. We will not go into the details of that. Autocrine signaling drives the androgen independent. Androgen means it's a male sex hormone is androgen, and autocrine signaling drives the progenitor cell proliferation at the bud tip. Okay, and remember this phenomenon is androgen independent. That means it can happen right from early stage of development. You don't have adequate levels of androgen in them. Much later after puberty only you will have that level. Paracrine signaling, that is autocrine signaling what we saw, which is androgen independent, that is progenitor cell proliferation at the bud tip of the prostate gland. And paracrine signaling elicits a complex transcriptional response determined by the development stage, which stage of development you are in, that exerts various effects on the epithelial proliferation and differentiation. So this is the paracrine signaling, because paracrine signaling can act a little bit away. Autocrine signaling is right on the spot in the same cell where it is being produced. In the prenatal prostate, that is very early development, during early development, hedgehog pathway activity drives epithelial proliferation and ductal growth. This is very early during the development. In postnatal prostate, that means post-delivery, postnatal prostate, what happened? Hedgehog pathway activity inhibits epithelial proliferation and ductal growth. So what do you understand from here? Same signaling event. During early development, it allows the organ formation normally present, but soon after birth, it do not want any more proliferation because it is done and dusted. The same signaling is doing the same job. You may have seen in some movies where the villain has paid money to the bodyguards of the rich man, and then the rich man thinks, oh, these people are my bodyguards, but the villain already paid them and they are now, so the rich man is now alone. So such kind of stories you would have seen. Same way, the hedgehog signaling is now, earlier, before birth it was a pro-proliferative, after birth it is not favoring proliferation, it is stopping it. So androgen independent progenitor cells generated by autocrine hedgehog signaling during development can survive castration, that means if you remove the organ, and enable prostate regeneration in response to testosterone supplement. So now you are talking about a third scenario. Prenatal, postnatal, we saw it. Now something happens because of androgen supplementation. That is why the levels of androgen, if it is very high, that can act as a condition, or not that anyone who has got a high level of androgen will end up getting a prostate cancer, not like that, but it will favor a situation because it is androgen dependent. So far you saw is androgen independent. Sometimes the hedgehog signaling can be facilitated by the androgen. That is why even if you castrate, there is a vulnerability of this proliferation of this gland possible because of the androgen supplementation. So this is what you can see here in this picture, how sonic hedgehog signaling happens, and the gli is the actual transcription factors and epithelial progenitor cells and mesenchymal cells, and how they are acting onto contributing to the proliferation. Red means stop, yellow means stagnated, green means go in terms of proliferation. So homeostasis and neoplasia, let us see. So epithelial cell, what it will do, it will release the hedgehog ligand, and there will be gli production. Gli is the transcription factor based on the hedgehog signaling in the stromal cells, and stromal cells will release a lot of paracrine factors, and it will allow the epithelial cell to proliferate. During carcinogenesis, what happens? This is homeostasis, normal scenario. And during carcinogenesis, what happens? Same event that is occurring, but however, these paracrine factors allow disorganization of this, the stromal cells and epithelial cells. Here it is well organized, here it is not organized. This is what happens. So the target genes are activated by paracrine Hedgehog signaling in adult prostate, and the phenotype of this stroma determines the effect of epithelial tumor cell proliferation. And in normal adult prostate and prostate cancers with a normal fibroblastic stromal cell, the target gene profile resembles that of the postnatal prostate. Postnatal prostate, proliferation is minimal, same signaling event, and the effect of paracrine Hedgehog signaling is homeostatic. It is involved in maintaining the homeostasis. In prostate cancers, let us see what happens. The so-called reactive myofibroblastic stroma, the target gene profile resembles that of the prenatal prostate. That means proliferative. Prenatal means proliferative prostate. And the effect of paracrine signaling is to promote epithelial tumor cell proliferation. So the autocrine signaling and the tumor cell have got a strong connection between during the homeostasis as well as in the neoplasia condition. So this is a schematic review of another tissue called urothelial cell. Urothelium is the lining of your urinary bladder, which is one of the simple organ as well as one of the tough organ in your body. Regulatory pathways and important for the urothelial proliferation, differentiation and regeneration upon injury. So you can see in this picture, so urothelial cells and markers. This is a typical urothelium. You have got a superficial cells here, and you have got intermediate cells here, and basal cells here, and stroma cells. It's a very simple structure. Dedicated cells are there because it contains urine. Your urine, which is a toxic hypertonic liquid, it has to resist the damage that can occur. So you can see here in A and B, it is urothelium and urothelial markers. As you can see here, different markers are there in different tissue types. What you are seeing, it is color coded here. You can see here stem cells, basal cells, intermediate cells and superficial cells. They are color coded. BMP4 is expressed in the mesenchymal compartment and the signal to the urothelium through its receptor BMP-R1A, and to regulate the proliferation and the terminal differentiation status. Estrogen can regulate urothelial regeneration by regulating the BMP pathway directly or indirectly by downregulating the cytokine interleukin 6, which is a pro-inflammatory cytokine. Retinoic acid and Wnt signaling govern urothelial regeneration by regulating genes such as BMP4, because BMP4 starts with and it will facilitate its own production. During UTI, urinary tract infection, what happens? Uropathogenic E. coli invade and rapidly replicate inside the superficial cells, what you are seeing here, blue colored one or the violet colored one. Then the host tissue responds by exfoliating the damaged superficial cell layer and recruiting neutrophils and monocyte macrophages to clear the infection. The urothelium mounts a rapid proliferative regenerative response to re-establish the barrier. So now if you see in this picture, you will know the stem cell is doing this much bunch of action. It has to respond to the Wnt signaling and BMP signaling. As you can see here, it produces these cells, stromal cells produces BMP4 and it responds to the Wnt signaling, and then it triggers in the stem cell, it triggers a sonic Hedgehog signaling, and as a result there will be production of BMP-R1A, that is its receptor, BMP's receptor, and it relay this information to the basal cells, and it will lead to the terminal differentiation and it will restore the superficial cells. This is the normal course. But when there is a damage, as you can see here in this picture, it must superactivate this pathway so that it can restore back the normalcy. So this is how, and your urinary bladder epithelium damages and it gets restored back. So the regulation of urothelial development, regeneration in cancer, the urothelium plays a critical role as a permeability barrier to cytotoxic factors on waste products present in the urine. The essential barrier is maintained by the specialized superficial P1 urothelial cells we saw in the picture. A dysfunctional response to the urothelial injury and dysregulated repair and restoration of the superficial barrier can lead to chronic bladder disease. this process of underlying urothelial repair complex involving structural elements and signaling pathways and trophic factors. So we'll study more about in future class. Thank you.